Hello, everyone. This is Clay Chase with Torrey Hills Capital. Welcome to the NervGen Pharma podcast for August 16th, 2023. Trust and hope you're all doing well and having a great summer so far. Now, as many of you probably know, the presenting company that we have on today is still kind of flying under the radar. That's why we're really excited to bring to your attention today. For those of you that are familiar with the company, this is going to be a great update. Lots to talk about. Some of you might not have met the company's new CEO, which is going to be our presenter today. So I'm really excited to have everyone on the call today. For those of you new to the story, you're in for a treat. This is really a great company. So what this company is bringing through the clinic, it's an early stage biotech company. They're bringing through the clinic a drug that promises to be truly transformative for a myriad of conditions that affect the nervous system. Now, we're talking about a drug that could not just stop the progression of these indications, but could truly repair the damage from conditions like spinal cord injury, MS, ALS, stroke, possibly even Alzheimer's. It's important to note that each of these indications that I just mentioned, for the most part, have no real effective treatment or standard of care, and they're all very high value markets. NervGen trades on the OTCQX under the ticker NGENF, and on the TSX Venture Exchange on the ticker NGEN. NervGen Pharma shares are currently trading around $1.30 per share, has a market cap of roughly $106 million. Now, with us today to discuss NervGen Pharma and their growth strategy going forward is the company's recently appointed CEO, Mr. Mike Kelly. Hello, Mike. How are you today?
Doing well, Clay.
Great. Okay, I'm going to stop sharing my screen, which should allow you to put yours up, and then we're very excited to have you on the show today and looking forward to the presentation.
Excellent.
And it's up.
Thank you. So I'll introduce myself in a second. I just want to let everyone know that I will be making some forward-looking statements in this presentation. All of the risk factors associated with the company and our company presentation are up on the website, as well as our annual information form and cedar.com. So my name is Mike Kelly. I'm the CEO of NervGen Pharma. About 5 months ago, I joined the organization. Let me give you a little bit of my background. I've been in the pharma industry for about 30 years. I started off with a company called Takeda Abbott Products 30 years ago on the commercial side of the business. From there, I went into smaller, earlier startup biotechs, companies like Pharma Incorporated and Guilford Pharmaceuticals, where I cut my teeth building out commercial organizations and launching several different products successfully, which built the foundation for me to be able to join a pure startup, a company called Azure Pharma, where I was a member of the founding management team. And we built that company from the bottom up, from 4 people up to about 100 people, and ultimately sold it on to a company called Jazz Pharmaceuticals. From there, I joined a company called Covis as my first CEO role, and we successfully built their portfolio and sold their assets onto a company called Concordia Healthcare in Canada for $1.2 billion. And then most recently, I was president of North America for a company called Adapt Pharma that developed and launched Narcan nasal spray. That was one of my most successful career steps, and one that I'll refer to a little bit in this presentation because there's a lot of similarities between what we did at Adapt and what we can do here at NervGen. So that's me in a nutshell. About 5 months ago, I was sitting in your shoes and I was approached by a company, a very well-known recruiting agency, and they asked me to take a look at NervGen. At first, quite honestly, I was a little skeptical. I went on their webpage, I looked at everything that they had posted up there, and they had this slide with a tagline, enabling the nervous system to repair itself. It was a pretty bold statement for a very small company. And as I mentioned, I was a little skeptical at first, but as I started to dig into the science and understand the technology, my my skepticism turned to intrigue. Then I met with several of the board members, and then Jerry Silver, who I'll talk about here in a minute. He's the discoverer of this technology. I started to get very excited. Then I met the management team and I was fully engaged because I knew that we had a platform between the science, all of the preclinical data, the board of directors and the management team, we had a platform that we could pretty quickly move this company from a discovery stage up through clinical and into commercialization. And I'd like to show you some of the highlights of this story. And I urge you, if you dig in and you really understand what we're all about, you'll get into the same place that I am. So we have some highlights. Our first drug, NVG-291, is a first-in-class drug candidate. And I'll talk a lot about that today. And it has the potential to allow the nervous system to repair damage, which is a first-in-a-kind molecule. It's demonstrated its effect in many different animal models of nervous system damage, and I'll walk you through a lot of those today. We are targeting initially spinal cord injury, but then several huge markets of high unmet medical need. And the most exciting thing is we just announced that we started our proof of concept study in spinal cord injury, and we got IRB approval last week, and we're expected to start enrolling patients shortly. This is the management team I referred to earlier. We all have 25 to 30 plus years of experience in the biotech and pharmaceutical industry. I'm not going to go through all the names here, but we represented, or our backgrounds represent several different companies. Dan Michael was with NGEN, heading up their field of neurology. He's the brains behind our clinical program, and he designed our spinal cord injury trial. He's also has a specialty in MS. Matt Lukachev is our VP of Preclinical Research, is also one of the founders of the ALS Institute. So we have a broad range of leadership and a strong foundation to build upon. Let me walk you through a little bit of the history of, let me start before the history of the company and the history of the technologies. I have to go back to the early 1900s to where the mindset was that the central nervous system could not repair itself. And that was a pretty common belief that everyone believed in, that there was no way that once damaged, the central nervous system couldn't repair itself until landmark discoveries made by Dr. Jerry Silver. So I met with Dr. Silver, and Dr. Silver, when he was teaching me about the technology, said that his focus wasn't necessarily on trying to get nerves to regrow, but trying more to find out why nerves didn't grow where they should have grown. And through this discovery, he discovered that there were these molecules called chondroitin sulfate proteoglycans, and they connected with cells and put a cascade of inhibitory events in place that prevented nerves from regenerating and regrowing. So it was a landmark discovery. Then for the next 2 decades, there were several different studies where when you take CSPGs out of the equation, you regenerated, you remyelinated, and you enhanced plasticity within the central nervous system. So those discoveries led to, in 2009, the discovery of a receptor called PTP sigma, which was the receptor where CSPGs were connecting to and inhibiting or cascading the events that allowed the inhibition of repair. So that receptor discovery was critical in the fact that in 2015, they started to target that receptor to see if they could find drugs that would enable this cascade to not begin. And in 2015, NVG-291 was discovered, and in 2018, it was in-licensed by NervGen, and that was the starting and the founding of the company. I'll walk you through how this works in a second, but first let me talk briefly about the product. It's a cell-penetrating peptide, meaning it gets into the cell. It's 35 amino acids in length. It's designed to cross the cell barrier. The route of administration is a daily subcutaneous injection, similar to insulin in the belly fat, and it's manufactured by chemical synthesis. I'll walk you through very briefly how it works. So if we say that we enable the body to repair itself, when CSPGs are present in either a damaged cell due to spinal cord injury or a CNS damage due to MS or Alzheimer's disease, a glial scar is formed. And in that glial scar are CSPGs. And those CSPGs inhibit three key repair mechanisms that the body naturally will repair itself: plasticity, axonal regeneration, and remyelination. So this is a cartoon here showing that a brain and a spinal cord where there's some injury. It shows the glial scar in the middle, and it shows the axons being broken. And then this one shows an axon where myelin, which is the green coating, if you can imagine the plastic around a wire in your household, if the plastic breaks apart, the signals from the electricity go into the wood and not follow along the line. So remyelination myelination is broken up, regeneration is inhibited, and therefore there's not an electric current going from the brain to the extremities. When NVG-291 is introduced into the picture, it enables the body, it enables these three repair mechanisms to occur. So there is plasticity, there's formation of these new connections to axons. There's remyelination, which is, all of the green covering around the axons, the fatty substance that protects the axons is regenerated. And then axons are regenerating and regrowing around the glial scar or through the glial scar to allow connections. So this is a transformative mechanism which will allow you to treat many different disease states. And in fact, this was one of the things I looked at when I joined the organization. Typically, you'll see a small startup biotech, when they take a product out of academia, they'll have one or two reports of this product working in an animal model, where NVG-291 shows across many different animal models, many different disease states that have been modeled out, that we can actually enhance plasticity, enhance repair through regeneration and remyelination, and also enhance recovery and function. I'm going to show you some of these studies specific to spinal cord injury, and one of the reasons why we chose spinal cord injury as our first area to get into in human clinical trials. All of these disease states, this is where we're going. From left to right, you'll see spinal cord injury. Most folks think of spinal cord injury as a small market, but I could tell you it mirrors most of the larger markets. If you look at the system cost of spinal cord injury, it's $50 billion annually. The lifetime cost for a patient who suffers from a spinal cord injury, and I'll get into that in a minute, is anywhere between $1 and $4 million. And this is a significant unmet need. There's no treatments currently available. But if you look at all the other disease states that we're going after, they're also massive markets as well. As I mentioned, there's no FDA-approved drugs that enable sustained functional recovery. And let me talk about these patients to humanize this affliction. It hits mostly men, 78% men. Average age is 43. The average age has grown over the years. It's roughly 33 last year. And it's caused in an instant by a vehicle or a vehicle accident or fall. In more than 70% of the times, that's what happens. And the person's life has changed forever. They're hospitalized for 2 to 3 months. They go through surgery. And these folks, this is really it. They go through rehabilitation after surgery, and they push themselves to regenerate movement. And that movement plateaus somewhere between 6 months and a year. And wherever they land, that's where they are for the rest of their lives. 18,000 new cases a year, just about 300,000 patients living in the United States with a spinal cord injury, and just under 500,000 estimated worldwide incidents. I mentioned that these folks have rehab at first, surgery and rehab. The first couple of months, it costs $200,000, but then depending on the age that they suffered their spinal cord injury and the severity, the cost can range anywhere between $1 and $4 million for the rest of their life. And in addition to the economic impact, there's many other different things that these folks have to deal with throughout the rest of their lives. I'm going to touch on some of the animal data that we've shown and why we're so excited about getting this product into clinical trials. So this is an animal model done by Dr. Rink. It's a study done by Dr. Rink that looked at rodents, and this is called a BBB scale, which is a scale of locomotive function. The gray area on the chart on the right-hand side is the treatment period. So what happens is the animals start out with normal function, then they're injured 1 week after 1 week after the treatment began. There is some spontaneous recovery. The blue line is the placebo-treated rodents. The green line is active NVG-291R treated rodents. And you can see that there's a dramatic difference in the amount of recovery. Now, I know that this is a graph and BBB scale isn't known by everybody. So let me show you some of the videos that came from this study and exactly what I mean from improvement in function. So the rodent on the left, you can see, is dragging its back legs. Its tail is down. It has no function. This is the placebo-treated group after the treatment period. And you can see that the scientists conducting this study has to help the rodent along the beam because they have no function of their back legs. The right side of the graph here, now I'll start this video, is a NVG-291 treated group, and watch the back legs and the function of its tail. To the untrained eye, it looks like this rodent is, fine and should be a 21 on the scale, but roughly this is a 15. But it's a dramatic difference between the untreated and the treated group. So this gave us a lot of hope that we have something. I show you another video here. This is on the left-hand side, same animal models, the same back legs are being dragged up the ladder, no repair. And on the right-hand side here, you'll see that the treated animal walks up the ladder with perfect function. These results are dramatic. These were never seen before prior to this clinical trial. And they gave us a ton of hope that we have the ability to translate this into humans. So that was acutely treated animals. So animals that were injured right around the time the treatment started. There's also a study done with chronically treated animals. And what we mean by that is the injury occurred 12 weeks prior to treatment starting. So there was some spontaneous recovery, as I mentioned, in both the placebo-treated group and the NVG-291 treated group. But you could see once treatment started, there was some separation and continued separation of improvement in function. Now, this is a different study. It uses 4-limb locomotive rating scale, which is a scale that looks at the digits or the fingers and the limbs of the rodents to see how well they operate. But this was a study that gave us hope that in a different population, the chronically injured model, that we would see hope even after the spontaneous recover period. The last piece of data I'll show you from a preclinical perspective is bladder function. And why is bladder function important? Because we recently did some market research that indicated that bladder function is one of the top 3 things that folks that have spinal cord injury would like to regain function of. It gives them independence, it gives them a bit of their life back. And this study basically shows void frequencies per hour in rodents. And essentially the takeaway of this slide is showing that as the dose increased, so did the void frequency increase. Now, typically it's a void frequency of 2 per hour, 2 voids per hour for the rodent. And as you can see, these doses, we didn't even get close to the doses that were in the 2 previous studies. This is about 1/10 of the previous study's dose. There was a continuous increase in bladder function as you increase the dose. So that gave us a lot of hope into getting into human trials. So we started our first phase 1 and completed our first phase 1 clinical trial. What phase 1 means is that you have to prove that your drug is safe in humans before you can get into proving that it's effective. So there were 70 subjects, both in a single and a multiple dose ranging arm. They were assessed through 8 days and 28 days in both arms. And the takeaway from this study is that the drug is well tolerated in all doses. None of the patients discontinued treatments. There were no serious adverse event. The only common adverse event seen was injection site reaction. And that's to be expected when you're giving an injection in the belly fat on a daily basis for that period of time. This is what we're most excited about. We have a 1b/2a proof of concept study in spinal cord injury. And what that is we're actually treating patients who have suffered a spinal cord injury. There's two cohorts, a chronic cohort, just like I showed you with the preclinical model, and a subacute cohort. So the chronic cohort is going to be patients who were injured 1 to 10 years ago. The subacute is going to be patients who were injured 10 to 49 days ago. It's a 16-week trial. Patients are going to be randomized into active NVG-291 versus placebo. They're going to get once daily injections. This is going to be conducted at one center, Shirley Ryan Ability Labs. Shirley Ryan Ability Labs in Chicago is ranked the #1 rehab hospital for the last 30 years. Monica Perez is our primary investigator in this trial. And the reason we chose Monica is because she is an expert in electrophysiology, and I'll talk about that in a second. The reason that we only are doing this in a single center is to decrease variability across centers to increase the ability of seeing a signal in these clinical trials. So there's two co-primary endpoints, hand and leg motor connectivity. And what does that mean? We're using electrophysiology, where you send electrical pulses from the brain down through the hand or down through the leg, and you measure amplitude of, it's a quantitative measure. It's MEP amplitude. It's motor evoked potential amplitude, which shows you the amount of electric current that is going through that muscle. So we can, over time, see if we can increase the amplitude or increase the amount of connectivity that the body has. So that's our primary endpoints. The secondary endpoints are going to be clinical measures. These are going to be based on traditional walking speed, hand function, and neurological assessments. We also have a host of other electrophysiological measures with different other muscle groups, and then exploratory endpoints, where we're looking at plasticity, quality of life, bladder function, as I mentioned earlier, and biomarkers. Some of the clinical advisors, these are the folks, the who's who of spinal cord injury in the United States. These folks helped Dan design this study. And this is our pipeline. We're excited to get the spinal cord injury off the ground. It's our big next inflection point as a company. We'll be enrolling patients in the next couple of weeks. We will complete the chronic cohort by the end of the year, and we're going to have a data readout of spinal cord injury in the chronic cohort by mid-year next year. So that's going to be a big milestone for the company. We've done preclinical work in stroke. We have fantastic data in stroke as well. And our hope is to not only continue stroke in 291, but potentially in NVG 300, which is our next generation compound that has come out of our discovery. And then we're also going to be getting into preclinical work in ALS, Alzheimer's, and multiple sclerosis. Our board is made up of several executives from the pharmaceutical industry. Everyone on this page is successful either in the financial community or in the biotech industry. And our value drivers over the next couple of months to years are the 1b/2a trial. We've received IRB approval, which is clearance to start the study. Our first patient is expected to be dosed in the upcoming weeks, as I mentioned. We have preclinical data in multiple indications that we're hoping to get read out in 2023 and 2024. We have a new compound in NVG 300, which is exciting. It allows us to put another compound into the preclinical and clinical space. And as I mentioned, our proof of concept readout mid-year next year. With that, I'll stop and open it up for questions.
Great presentation, Mike. Really like the videos. It shows how well the drug could work. Certainly shows how well it works in rodents and animal models. If you have a question for Mike, feel free to chime in here. You can either click on your Q&A icon or your chat icon. You can raise your hand and we can ask your question right online. And while we're waiting to see if we have any questions, I do have a quick question. Now, and I don't know if you can answer or not, but in the spinal cord injury model, you're going to test for both an acute and a chronic type of patient to see if the drug works if they've had the injury for a while. Do you know if that could be the same case for stroke? Because I know a lot of people, if you don't get the stroke right away, the chances of having some efficacy are limited. Is there a chance that this could be a stroke patient who's had a stroke for a duration of time, 6 months or something like that, to go in and repair some of the damage? Or is it just too early to talk about something like that?
No, we believe that the way the mechanism works, I mean, if you think about how the body repairs itself when there's CNS damage, if you go back to the cartoon of the glial scar, the body will attempt to try to regrow and remyelinate. But the glial scar, the inhibitory effects of CSPGs in the glial scar are so potent that it does not allow reconnections or connectivity. We believe if the proof of concept in the spinal cord injury community in the chronic cohort is positive, then that's going to be a mechanism to show that even year after the connections had been stopped, that you can actually restart those connections. And we believe in our stroke data that's up on our website, our preclinical stroke data, we have cognitive impairment that was improved. We think that we don't have to be like the other stroke treatments that are within days of stroke, that you can allow some recovery, but obviously we have to bear all of this out in our clinical trials.
Great, makes sense. Excellent. If you have a question for Mike, feel free to join in. I've got a couple of quick announcements. If you'd like to get additional information on NervGen, you can do that on their website. It's www.nervgen.com. I think this presentation's up there. You can actually go and look at the videos. A lot of information about the drugs, the 2 compounds that they're looking to put through the clinic, as well as a kind of a timeline and a milestone chart of some of the things that we expect, or the company looks forward to having occur over the next, 3, 4, 5, 6 quarters or something like that. Also, we'll be sending out a feedback report. I've got a question here for you, Mike, and it is, what is the case model for recovery for a youth versus an adult? It's a good question.
I'm trying to, I don't really understand the question fully, but I'll speculate on what they're meaning. Is there a difference between youth recovery versus adult recovery? And our belief is that once somebody hits maturity and their nerves and their CNS is pretty much set, then obviously during early childhood years, your CNS is developing, but once you're fully developed, we don't believe that there's going to be a difference that you'll see in NVG-291 or the effect of NVG-291 in say mid-teens to adults. So we think that we're able to cover, especially in spinal cord injury, the entire population. So the 300,000 cases that are out there, I'm not exactly sure how many are in the early stages of youth, but with the average age being 43, we think that the market opportunity is going to be pretty much for the entire population.
Great. So just to be clear, when you said in adults you wouldn't see as much of an effect, do you mean that it's not a difference between how the drug works in adults and younger patients? But you
I think
adults, right?
Yeah, what I meant is once the CNS is established, once your CNS is mature, there should be no difference in our ability to enhance repair or enable repair. Not that there won't be an effect in adults and there will be an effect in youths, that the effect will be equal in both adults and youths.
Got it. Yeah. Thanks for that clarification. Great. So the code word today for your feedback report is NVG-291. That's the drug candidate they're putting through the clinic right now. That's NVG-291. Okay, I think we're kind of up against our time limit here, Mike. So I want to thank you so much for coming on. Any final comments before we wrap this up?
No, I really appreciate the opportunity to speak to you folks today. I think it's probably going to be the most exciting year since the founding of the company in the next 12 months. We believe that once we start to get patients enrolled by the end of this year, we'll have the chronic cohort done. And then, we'll have a readout of proof of concept data, and we'll be off to the races from there. So thank you.
Great. Well, thanks. Sorry about that. We're looking forward to having you on again real soon, possibly when some of these milestones cross. And I want to thank you again for coming on. Thanks everyone for coming on today. Like I said, if you want additional information on NervGen, you can go to their website, nervgen.com. You can also email me if you have questions for Mike. I know Mike's going to be out in the New York area here in a few weeks. So if you're interested in meeting directly with Mike, if you have some either clients or someone you'd like to have meet Mike, send me an email and we could try and arrange that meeting. Also, this presentation is going to be pushed up to our YouTube channel. So if you have associates or clients who would like to view this presentation, just send me an email and I can send you the link. Okay, great, Mike. Thanks again for coming on today. Really appreciate it. We'll look forward to having you on again soon.
Thanks, Clay. Take care.
You bet. Thank you. Thanks everyone for coming on. Have a great day, and we'll talk to you soon.